welcome everyone to the new edition of the EMBL Genelia bioimaging seminar series. And I'm very happy that today we have actually two speakers, Theodore Alexandrov and Volker Hilsenstein, who are both from EMBL and who will speak about spatial metabolomics, both from, I guess, a microscopy, but also image analysis point of view. And I want to first introduce Theodore, who's going to be the first speaker. And so Theodore Alexandrov is a team leader at the moment at EMBL, which means he's both the head of the metabolomics core facility here in Heidelberg, but also a research group leader. But he also shares additional appointments, for example, with the Molecular Medicine Partnership Unit here at EMBL and the Heidelberg University, and also with the Bioinnovation Institute in Copenhagen in Denmark. And so before Theo joined EMBL in 2014, he did a PhD in St. Petersburg and then a postdoc in Bremen, where then later on he also became a research group leader. But now we're very happy that he came to EMBL, and he's been here, as I said, for quite a number of years. And during this time, he really has pioneered this new field of spatial single-cell metabolomics, which is also the topic of the current seminar. And we're very happy to have you, Theodore, and please go ahead, share the screen, and we're looking forward to your presentation.
Yeah, thank you, Robert. Thank you, everyone. It's actually me who is very happy here to present today. And in particular, because actually my first postdoc was on image analysis. So my background is in, PhD was in mathematics and statistics. Then I moved to Bremen, Germany, and I did time series analysis. But then I actually got an image analysis. And then specifically, I worked at the University of Bremen in a group which was focusing on inverse problems. So there was a lot of hype back then about inverse problems and how they can be used for image analysis, in particular for image correction, because, you know, there was all this this thing with Hubble, right? When the knowledge of the function and how the function contributed to the blurring of the image, and inversing this function helped actually in increasing the sharpness of the images. And there was lots going on back then. So that's why then afterwards we moved into, I kind of like transitioned a bit more towards omics and even doing supervising some experimental work. But I still very much enjoy image analysis, and it's very dear and near to my heart. So very happy to present some of our work here. So let me share my screen. Do you see my screen?
Yes, looks very good.
Yeah, wonderful, thanks. So, okay, my name is Theodore. Indeed, I work at EMBL here in structural and computation biology. And also, since this year, we have the second lab in Copenhagen in Bioinnovation Institute, where we actually commercialize some of the developments in single-cell analysis. And before I start, I actually want to acknowledge and appreciate and express my gratitude, but also excitement to all team members that we have currently in the team. But, you know, the work which is here, it's really that I'll be presenting next. There are even more people who contributed. And another aspect of our team is not only these colorful images, but if you start looking at, they actually combine a lot of different expertise in different fields, combined from biology. And this is normally where the questions come from. It's great to collaborate with people, and we collaborate a lot, but it's great to have experts in-house, you know, who can really make sense out of the data that we generate. And the data that we generate with mass spectrometry. And you will see some of the, you know, even photos of what mass spectrometers are. And here it's so great to have experts in this field. So because of my background, but also because mass spectrometry generates a lot of data, we need bioinformaticians, computer scientists, system biologists. And here they contribute to really kind of develop algorithms, but making sense out of this data. And our team would not be where we are right now without software engineers. And here we have 4 devoted software engineers, Lucas and Sergey. They work on cloud computing. I'll show you next. And today we'll see actually a presentation. I'll give just an introduction, but the main presentation will be by Volker, who is image analysis specialist, and which was prepared together with Andreas, who is software developer, and they together, they developed the software that Volker will be showing, which integrates a lot of data analysis and image analysis and so on. And for Volker background, I'll introduce, and Andreas' background was actually in chemistry before he moved to the career of software engineer. So long story short, it's very cool and interdisciplinary team, and I'm very happy to work with all of that. Now moving towards what is our research focus, and here, so I'm depicting here. So it's about metabolic cell state, and so definitely in imaging, but also in single cell analysis, such as single cell RNA-seq or transcriptomics, we now know that our bodies, they consist of multitude of cell types, where cells, they differentiate and they, like into different cell types. But what's even more interesting, what happens with the cells? False differentiation. And one cell type can have multiple functions. And these functions are tightly linked with which small molecules or metabolites inside the cells and metabolic programs. And what's also very interesting, that metabolic cell state is a very dynamic phenomenon. It can change within sub-seconds. So how does it change, and how our bodies, they really regulate the metabolic cell states to be in homeostasis? This is the key question. We know that metabolic cell state is controlled by DNA and cellular programs, of course, but even more interesting, how it's actually controlled by non-genetic factors. And this is what metabolomics is about. To capture this input of this and profiles of non-genetic factors, such as neighboring cells, environment, we can think about tumor microenvironment, we can think about climate change, increase of temperature, like really global environment, disease. Reprogrammed metabolism is hallmark of cancer and other diseases. And how can we build therapy that would capitalize on the knowledge of this metabolic cell state? So we do it, in particular, over the past years, by developing methods. We are mainly technology and method developers team, which methods for spatial and single cell metabolomics to provide readouts in the native context, to resolve heterogeneity, and provide these readouts which are closest to the phenotype, these metabolic profiles, small molecules, lipids, and other metabolites. So how would we do it? We use mass spectrometry for detection. I'll introduce this technology. We also, and this is really absolutely essential for our team, is to really develop big data software which can handle big data, but also, as you'll see, visualize and share this with others as well. And then also more and more we're getting into coming back to sort of my roots. I started this image analysis, I started in machine learning, but then we get into omics, but now we're getting back to machine learning and AI as well. So this is the overarching focus of our team. Now, why do we need spatial single cell metabolomics, and what are the potential applications? So one application, of course, is tumor microenvironment. We know that it is built of cells of different types. Here we see cancer cells depicted here. Here we see tumor-associated macrophages, immune cells, which actually located here. Here we see cells from adaptive immune system, T cells, B cells, and many other cell types. And they actually reside in their specific metabolic cell states. But what's even more interesting, and what's not possible to detect, say with single cell RNA-seq or spatial transcriptomics, or sometimes even with imaging, is that here it's very special chemical environment. So here we have the gradient of acidity. Inside, the solid tumors are more acidic because they secrete lactate. So it's also hypoxic environment because there is not enough vascularization. There is less oxygen here. So there is all these differences in chemistry inside the solid tumors, which can be studied with spatial metabolomics. And the second application that we are very interested in is how immune cells, and here shown in green, they get attracted here into the solid tumors. They get attracted here by special molecules called chemoattractants. And then they squeeze through this blood vessel wall, and they immediately get into like completely foreign environment. So here from really nutrient-rich blood, they get into this hypoxic, acidic, and very competitive environment that they need to reprogram their metabolism to proliferate, expand, and also start killing all the cells. So how do they happen? This is absolutely paramount for understanding physiology, but also for us to develop therapies, in particular immunotherapies. Okay, these are two applications that we're interested in. Now let me show you photos of our instrumentation that we have in the lab and we use for spatial and single cell metabolomics. So this is called imaging mass spectrometry. It comes in different flavors. We use MALDI imaging mass spectrometry. MALDI here stands for matrix-assisted laser desorption ionization. So this is a mass spectrometer here, this gray and blue box, coupled with laser, which provides spatial sampling. Here you see our scientific officer Prasad, which takes a tissue section here, which is on the glass slide. which places it here, then afterwards puts it into this chamber, closes the lid, and what happens next is the laser will start shooting at this location, then moving the stage in this location, moving the stage next location. And when laser shoots at a particular location, there is a sort of micro explosion happening on the tissue surface. Molecules, they fly off, they get captured by mass spectrometer, by vacuum, and they get actually detected there, so that from every pixel, we're getting this mass spectrum, which is a very high dimensional vector. And for every dimension of this vector, we have number of molecules which correspond to this dimension. So you can think of this as sort of quantified barcode of which molecules were at this particular location. And now, after we repeated this in a rasterized manner, we can actually select a particular dimension here, which corresponds to molecular weight of a molecule. You can map intensities across all spectra towards their initial locations where they were sampled. And now what we are looking at, we are looking at relative abundances of a molecule of interest as this, what we call ion or metabolite image. And now through the process of metabolite identification, we can find which molecule is hidden or represented in this image. So the technical characteristics of this technology is that spatial analysis can be done with a 5 to 10 micrometer pixel size. And there are already prototypes demonstrating 1 to 2 micrometer pixel size. So pretty close already to light microscopy spatial resolving power. So we can detect a lot of molecules in one shot, more than 100 molecules, metabolites, lipids, also drugs. This technology is heavily used in pharma. So, but it also generates a lot of data. Why? Because we have a lot of pixels, and per pixel, we have a spectrum which can have more than 10,000 dimensions. And these dimensions are even different between different spectra. So it means if we take unique dimensions, we can have up to 1 million of channels or dimensions per data set. Now you understand why there is a lot of data. So, and over the past years, this technology became the major technology for spatial metabolomics. So we have contributed to this technology by developing cloud software called Metaspace, where people can put their data in, it processed on the cloud using high performance computing on Amazon cloud and IBM cloud. They get their molecules back. They also share this data publicly, and this project is supported by European Commission National Institute of Health. So how it looks, if you go to this project, which is open source and free for everyone, metaspace2020.eu. If you go there, so there is thousands and thousands of data sets. Let me show you just a few. What we'll be looking at, whole body sections through a mouse. The data was generated by 2 pharma companies. I'm picking here a data set which was uploaded by the company Servier from France, pharma company. And what we'll be looking here on the right, this is actually was a section through the whole mouse, adult mouse. And you see here optical image, but on the left, you see here images, molecules, which were detected here. And now I can click at the molecule because everything is already processed. I can see the structure of the molecule, localization, and so on and so forth. And it's everything at the fingertips. And I can associate it with anatomical structures and so on and so forth. And we have not only one data set, but we have 6,000 public data sets in this platform. And it's growing and growing again, because it's open source, it's community supported, it's very collaborative, and there is a lot of publication already coming out of this platform. This is about spatial metabolomics. Now the question is, I would like to explain you how can we do single cell analysis and set the stage for Volker presentation, how we do it. So putting it into perspective, first of all, single cell metabolomics is a really emerging field. Just a couple of years ago, there were just 100 publications on PubMed on this topic. But right now it's exponentially, as other single cell analysis is really exponentially rapidly developing field. So if you're interested, last year, single cell metabolomics was recognized among top 10 emerging technologies in chemistry by IUPAC, which is a standard for Society for Institute for Standardization and Union for Standardization in Chemistry. And if you're interested to learn more about single cell metabolomics, you can check out this technology feature in Nature Methods, where they discuss technology, but also applications. Very easy reading, just a couple of pages. And now the main slide of my part of the talk is that, so we have this method, and this method called SpaceM, and this is the method for spatial single cell metabolomics. And I would like to present you this method, but first I'd like to tell a story, because when I came here to EMBL, we didn't do single cell analysis, we didn't know single cell metabolomics, but then PhD student Luca, he joined us, he was the first PhD student in our team, And he wanted to really develop some ambitious methods. And there was single cell already RNA-seq going on. And he's like, hey, can we do single cell metabolomics? And at that point was really unclear whether even sensitivity will be sufficient. But he decided to try it out. And this worked in marvelous way. And he really set the stage for the research program in our team. And now our team, you've seen about 20 people, and almost everyone does something of single cell RNA, single cell metabolomics. So how does the method that Luca developed and we perfected afterward, how does it work? We take cells, we put them on a glass slide. This can be cell lines, this can be primary cells. We even now work with the cells dissociated from organoids. And then once we have them on the glass slide, we do microscopy. And here we do it for two purposes. First, to find where the cells are, to find cell outlines, but also to find their phenotype, in particular fluorescence phenotype and morphometric phenotype. Then what we do next, we actually do this multi-imaging mass spectrometry to find the pixelated images of individual molecules across this field of view of cultured cells. So we run them through this cloud software to find which molecules are hidden behind these images. And then what Luca has developed is a way how to overlay computationally these two types of data, microscopy and imaging and spectrometry, on two different spatial scales, right? So how to overlay it with a subcellular precision. And once we can do this, we can start assigning signals from pixels to individual cells. And what Luca has developed also a way to normalize and integrate all this. And now we can assign normalized intensities from the pixels to individual cells. Now we have single cell data. And we can also summarize it in spatial molecular metrics, where for every cell we have an image. We can go back to this microscopy. We can find the image of the cell, but we can also have quantified intensities of metabolites. And they can be here about 100 metabolites or several hundred lipids and across all cells. Then we have fluorescent intensities here. For example, this one, this can be staining, can be reporter. It can be even immunofluorescence. Morphometric properties, elongation, and others, which can be quantified with for example, cell profiler, but any other software. And spatial features with which cells are next to each other. And this is the foundational data that we get out of SpaceM, and we can use for a variety of analysis. If you're interested to see how we applied it to a non-alcoholic steatohepatitis, you can check out this paper. But we have many, many more projects now going on. So, but with this, I would like to wrap up my introductory part. I would like to, before I pass it to Volker, I would like to acknowledge everyone who contributed here, our current team members, our lab coordinator, Shiraz, developers of the method, software developers, Andreas and Volker, data analysts, and many, many more people, the selected past members, in particular Luca, who developed the methods, collaborators, funders, of course, from different countries, from Germany, from US, Switzerland, and Denmark. And with this, I'd like to pass it to Volker. And Volker, as you have seen in my first introductory slide, he combines the expertise in software and in computer science, actually in image analysis. And I know Volker from the past because when I joined EMBL, he was the person who was helping lots of people here at EMBL to really set up methods for microscopy and really help with image analysis. And then I knew that he moved to Australia, and then we opened position for a scientific software developer. He applied, and we were very fortunate to recruit him. And he combines the expertise of image analysis and also microscopy. And as you will can learn about him, that he actually throughout his career he spent some time in Australia, in beautiful Queensland and Brisbane, but also he has roots in Heidelberg, and now he's again back to Heidelberg. And we're very, very fortunate to have him here, helping us develop the software, and in particular, bring in this community aspect using tools like Napari and so on. And with this, without further ado, I'm passing it to Volker to introduce the technical parts of the software for SpaceM and also aspects of image analysis. Volker, over to you.
Okay. Thanks for the kind introduction. I will go very much more into technical details. Share. Can you all see my screen? So let's start with the slideshow.
Yes.
I will also acknowledge Andreas, who is in the audience. I mean, this is really equal parts of each of us doing this. I've got, this is what Theo just explained, how you raster with the laser across a sample to get spectra and then do annotations. The point why I bring this up here again is one thing that you need to do before you do this. You apply a coating on your sample, and that there's varieties of coatings, and that coating can actually, at least for microscopy, almost obscure your cells. So that is relevant for the registration after. You can't just use the image without any further tricks to register them. This is where Theo left off. I'll go into a bit more detail to see what image analysis steps we have. This is sort of a toy example, how you would do single cell metabolomics with Luca Rapes's method. On a little toy example where you just have 5 by 5 pixels or whatever, 6 by 4 and a few cells. So you would go to the microscope and take an image before you do any mass spec imaging. Because in the instrument, it's a bit hard to navigate with the sample. People have these felt marks, felt pen marks, and they also are useful to then register after you do the coating. So what you see with this coating, you have this motley structure in the light microscopy. It's been at the mass spec at this point, and you see the laser marks. They don't have a very nice PSF, but they're on a regular grid. You get the ion images. They will have been annotated, hopefully, at some point with metaspace. What we do is cell segmentation to get the individual cells. We detect the laser ablation marks, and we register before and after, so we can place the laser marks exactly on where the cells were, which we have at this step here. Then we can assign ion images to cells to get the metabolites for each cell. The other thing we do is, as Theo mentioned, we use morphological analysis. We do morphological analysis on the cells. How do we implement this? I'll go into a few of the other details later, but for cell segmentation, we just now use CellPose, which is from Janelia, so thank you very much. It simplifies our life tremendously. The cell features, we just use SK image region props, so that also makes things very simple once we have the segmentation. And the annotations we get from metaspace, as Theo mentioned. So the other effort is, you saw this toy example, and when I arrived, people did single experiments, but because quantification varies a bit, you have quite a bit of batch variation. There's many factors that influence your intensities, for example, that matrix spraying that you do. So it is desirable, if you want to do comparative studies with different treatments, to have multiple treatments on a single slide, as you would do in high throughput microscopy. We're aiming to do that with single cell metabolomics. So this is something that a colleague, Sharat, is working on at the moment. He's doing some CRISPR knockdowns or CRISPR kind of modifications to some cell lines. I'm not exactly sure what he's doing. As a format, we're using regular slides, but to see the different wells, we have these ProPlate things. So there's this silicon layer and a plastic thing that clips on. And I replaced the felt pen marks because I worked previously at EMBL, and I knew that there was a more or less unused microdissection laser dissection microscope sitting around. So we're making these custom slides for this process. Okay, so we want to move to high throughput, and we want to make this all robust software, not just a bunch of Jupyter notebooks and scripts that are error prone to reproduce. So we want to make it really so that all of our lab members and then beyond can work with this software. To do this, both Andreas and I, we like to work in the Python ecosystem, and we trying to use modern tools. So one component that is really essential to what we're doing is the next generation file format that's based on ZAR. Why do we need this? So it's chunked. So we have little individual pieces stored on disk, which supports lazy processing or out of core processing. So we can process bit by bit without kind of decompressing the whole volume. We can just decompress a chunk if we have compression, and we can access things. We can store pyramids. We can also store our segmentations. And because we are dealing with registration and several images, we are also using transformations and collections. So you have several images in one file with affine transformations. And this is something that is still there, where the specification process is still ongoing, but Andreas made sort of an implementation for us, not to preempt the specification, but just to have something to work with. The other thing is napari. During my time in Australia, I worked with Juan Nunes Iglesias, who is a very close collaborator, and he is one of the co-founders of this project. So I kind of saw this from the early stages, and as a Python aficionado, it was nice to see an alternative viewer emerging. We use quite a few features from napari. So we use the ability to view multi-scale pyramids, which we store in the ngff. Napari supports lazy loading of chunks. So if you have a very large data set, you only load from the disk what you're actually currently looking at. And it's got this concept of layers similar to Photoshop. You can blend them and superimpose them. And we also have specific layers for shapes and points with metadata, which is very useful for us. Then another thing that plays nicely with chunked storage is dask arrays. Basically, they are arrays that also are chunked, and dask also has like building a compute graph. So if you work with arrays, you don't have to process everything in the moment you execute a command. It's like you build a graph first, and then when you, at the end, you compute something, and then it only picks the chunks that are on the other side of the graph. So if you just have a small section, only those chunks are loaded and displayed. So that is sometimes called lazy computation. Another thing that we then do, maybe if I go back to napari, one of the reasons we use it, we have a few interactive steps that require user input. But we don't want to have user input for 40 wells. So the idea is to have the interactivity at the beginning. The user can inspect whether everything is right with the processing parameters they choose on some tiny sections. They can do a few clicks to do a course registration, and then we do the batch processing based on some config files that are generated, and that ensures that we can reproduce that same run with the same parameters based on the configuration files. I won't have time to go into all the tools we developed. I'll try and show some of them here that I highlighted. In a sense, this is the simple workflow that I showed earlier, but with the tools that we developed overlaid. The first thing is building, we're scanning whole slides, and unfortunately, there isn't really, I mean, whole slides have been around for a long time with pyramid formats, but there isn't a lot of good open source tools to build them. There's Ashlar now, which didn't satisfy all of our needs. There's some other things, but they don't necessarily write pyramid files unless you have enough memory to fit the whole image into your memory. So we try and address this in our group with this tool. We get mostly files from a Nikon microscope as ND2. Yeah, we used this tool to generate a preview of the whole image. but we can use napari to represent these layouts. So you can just drag and drop them from JSON files, and you have the layout as a presentation. And then we can generate a preview, either in full resolution or with arbitrary subsampling. And then we just have a very simple 3-click registration. So you pick which some corners of this known layout, click the corresponding points, and it will deal with things like, has the user put the slide in upside down, right side up, left way around, all of this sort of stuff. And then we already have it more or less in our slide coordinate system. We also have built-in flat fielding support. Some of the pictures you see that I'll show later haven't been processed with flat fielding support, so you still see gridding. But for people who also want to do quantitative fluorescence, so this is an essential step. Yes. Finally, when we build our whole slide images, we don't do yet registration yet. That's purely based on the stage coordinates of the metadata. So we just place everything where the microscope says it was, without fine-tuning based on correlation. For this, we currently still rely on Big Stitcher. So from this tool, Big Stitcher projects are generated and automatically in the background. Big Stitcher is launched, it will produce some intermediate files, and we also convert them to ngff. So this is then for individual wells. And we do that both pre and post multi, and all of these end up in one single ZAR file using the collections and the transformations feature that Andreas implemented. And so the transformations don't transform the actual data. They basically just tell the viewer or other software where in the world coordinate system do you have to place this tile. I mean, it's not rocket science. There's other software that does this sort of concept, but it's just very convenient for us. And when we in later stages do fine registration of the pre and post multi, we don't transform the images. We just update the affine transforms that we already have in there.
Volker, what do you use to open the ND2 files in dask?
I will come to that. I use a library by Tally Lambert. I actually have a special thank you slide for Tally Lambert at the end. I mean, he's been basically implementing tons of the features that we use in Napari, and he also has a reader that takes ND2 and directly gives you a dask array. So you have the lazy loading. You don't read everything from your ND2 until you need to. Yes, but that hopefully answers the questions.
Thanks.
To go, I haven't said how we actually go and get this large preview image. So we also use dask for that. I wrote a little blog post on how to do it. We haven't actually released this SpaceM stitcher as fully open source, but individual parts. I also had some help there from Marvin, who's and Genevieve, who both work on dask image. So if you just have image tiles and you know their position, you can place them according to the stage thing. But in this toy example, it's probably only 4 or 5 gigabytes. You could easily just do that in RAM and save it. But if you really want to go to, say full slides captured with 40 times objective or something, you might have 600 tiles, and that for several channels, that quickly becomes a problem to do that in memory. So you want to do out of core writing and pyramid generation. So with dask, you can do this block processing. So we have these chunks. We find where would our tiles intersect chunks. And then we use map blocks, so that we have a fuse function that we define here. And basically that is called for each block. It also gets some data type from the intersection. So when a block is processed, it's sort of indexed by its coordinate. And then for each of these chunks, we have a list of Here in this example, it's file names, but now it's indices into a dask array with their respective affine transforms. And the fuse function doesn't do any more than basically loading the tiles that are specified and placing them within an individual chunk and blending them. And so I can run that on my laptop. I can also run it on a much smaller laptop that will easily work, on an 8 GB laptop. And you can create then ZAR. Once you've got the dask array, you can write it to ZAR. Dask itself doesn't write pyramids. Again, Andreas's implementation of ngff writer can take a dask array, create the pyramids, and write it to ZAR. Okay, then we have these preview images, and we found that people spend a lot of time at the MALDI instrument to navigate around and placing their regions of interest, which wastes a lot of instrument time and is also inconvenient the way it used to be. So now in Napari, we can do this thing where users just basically use the layout. They can deactivate wells that they don't want to, and then they can specify how many MALDI points they want, define some regions. You have region anchors that you can move around, which is happening here. So you can place them where you have your actual cells. When you've done that, you generate the MALDI points. If you don't want all of your MALDI regions the same, you can just go back to the Napari ecosystem and use the properties viewer, and then you can modify your region anchors before you actually generate the points. Then you go to the mass spec. Theo already showed a picture. It's got a XYZ stage, which is very precise, but the camera in there to actually view where your sample is very blurry and not very precise. What I added to kind of go from those planned regions to the actual thing is a little stage registration. So we now drive that whole software from Napari. So you connect to the MALDI software via Telnet. You can move the stage around. And similarly, as we did the registration with the stitching, you can click 3 points on that known layout, and then you have an initial registration. It also does a focus plane calibration. Once you have 3 points, you can just navigate around the slide by clicking into it or just selecting other well corners, and you can easily add 20 additional reference points to make the calibration better. So yeah, and then once you've done that, you load your MALDI regions that you have defined before, and you click on start, and then you see the progress where it's currently imaging. You can have that, overlay that on your preview, or here I already reused the slide that had already MALDI done on it previously for testing. Here, I mean, this is partly my cell phone camera, but you can also see, you might see that the image quality that you get within that chamber isn't very good, which makes the registration a bit tricky. Yeah, one step is now we actually know that where the things are. So before there was a segmentation step for the detecting the laser ablation marks. Now we know where they should be. So we can initialize a grid fit in Napari on the locations where they should be. And then the users can interactively fix it. They can review whether it's accurate enough. And if it's not, they can basically use a point and drag it. Again, that is some wizardry implemented by Andreas. When it's tricky to actually know exactly where your cells are, you can already download your annotations and sort of overlay it with the pre-MALDI image to help with this grid fit when you don't see the laser ablation marks nicely. Okay, I probably only have 5 more minutes. Once you have done all of this, you've got your pre and post multi-microscopy. I didn't show how you kind of do a bit of cell post parameter tuning. We have everything in place to start batch processing. And we use this prefect server, which builds a dependency graph, or a bit like in KNIME, but you don't build it with your mouse. You kind of build it with Python code. And once one processing step is like here, once all of the upstream dependencies of that processing step have finished, it will start running on that, and it will distribute it to different cores. You could even distribute it across a cluster if you want to. And then you have a web interface where you can see, you know, I mean, each task is one well. Back then we had lots of tasks failing. That was still when we were experimenting with it. Hopefully now that you will see more green. But if something fails, you can restart it. You can inspect the errors. Whew. And finally, we have a results visualization. Andreas has done that. I've got a slide for that, but I think I will skip to try and do a live demo. Hold on, I'll just get out of the presentation. And there's two things that I wanted to show. So, oops, maybe not all the way full screen. So this is one of the slides with the preview. So you can see the preview is already pretty good. What really helps us is this layer mode. So if I go into this preview layer here and change the opacity, I can go and check the pre and post multi. Let's maybe zoom out to where we actually have a multi-region. You can see the fluorescence channels. You can zoom in. This is not even a full resolution preview. Here you see some stitching errors. That's just because we just place it there. I mentioned I only show this for one well here. We then overlay this microscopy ZAR, which has the individually stitched images. And this one is then also registered more accurately. And I'll show this here. So here, if I zoom in, you will see this is pretty much perfectly aligned after our registration. And I'll show you this layout. You can just drag and drop it into Napari. You've got your layout layer here. You can attach properties. So in the end, well, in the future, we will want to have metadata for each well, like the treatments. Let's reduce the opacity. And you will also see this is the actual region anchor. So here you can see this is not super accurate. This is still problems with registering to the multi-stage. We hope we can increase that accuracy, and these are the points. Once we have higher accuracy of registration with the multi-stage, you could actually do things like that. You can actually, you know, I mean, you wouldn't do it by hand, but you could actually place your multi points on the individual cells and not deal with all of this post multi microscopy and matching ablation marks to cells. So if you can get the stage resolution there, you can simplify that workflow tremendously. I hope the lag isn't too bad with the Zoom. The other thing I mentioned is Andreas's results visualization. So one of the results, some of the results that we have are of course segmentation images. So you can use the labels layer in Napari. So here you see segmentations, but then if you go back to the beginning, we had these things where you calculate morphological properties. So you might actually want to see what is the cell area. So this is a bit unfortunate with this example because these are T cells, they're all round, so you don't have huge differences in cell area, but you can, maybe if I up the contrast, you can sort of, oops, now I saturate the contrast a bit there. But you can do heat maps for all of the morphological features you derive. this would be more convincing if I did it on HeLa cells or other cells where you have other phenotypes. And of course, we can do the same for molecules. Pick a molecule and have it visualized as a heat map on top of your segmentation results. So now we're looking at one well, but we have a dataset selector. So the dataset selector helps us dealing with whole slides. We might have 40 wells and the layers become too many in napari to handle. So this is a way to basically select one thing, but here it's not with flat fielding, unfortunately, but now you can use this observation, for example, this feature here, the molecule, or I'll switch to the cell area again, because I noticed we have a gradient of cell area across the slide. So if I amp that up a bit, you can actually see that the cells here in these wells are substantially larger than the cells here on the left. If I were to zoom in, you can actually verify that that is true, but you see it nicely with the heat map. So you have this whole overview of your experiment. I'll conclude the live demo with that. Just don't rush too much. Where have I got my presentation window? There it is. Let's go back. Lastly, with Python, you sometimes have the issue of deploying stuff to the users, especially with large data sets. I'm gonna go view slideshow. So our whole software, all the packages are built with continuous integration. Once we have the Python packages with continuous integration, we actually build complete desktop environments that you can start from a Jupyter Hub or just with Docker. And our users can log on via the browser. They have the EMBL file share mounted. They have GPUs. They have plenty of RAM to run this. So it's on the same network. Everyone has the same identical environment. So we don't have to, deal with people's Conda environments on their laptops or otherwise. And you can just deploy it to, like we have an individual workstation that's very powerful, or we run it on the EMBL Jupyter Hub. And if we're not happy with that, we can just deploy it on some other cloud service. And that, is nice for us rolling out new versions. So this is, how we do that. And the nice artwork is by Andreas. Okay, live demo we already did. Theo already acknowledged the team. I can only acknowledge Andreas once more. I mean, his software engineering skills are far beyond mine, and we wouldn't be there without that skill. And also Tally and the Napari developer community. I mean, Napari is really the tool that enables that, and Tally stuff. I mentioned Napari, Magic GUI, the ND2 library. When we create these big stitcher projects, that's a library called NumPy to BDV by Nikita, someone from the former Privish lab. I don't know his last name, unfortunately, but that is also a fork of something that Tally did. So I don't know whether there's just one Tally or many people under that name. That, anyway, I'll end the thing here to leave time for questions. Thank you.
Great. Thank you very much, Volker, and also Theodore for a very great and also insightful presentation on this new imaging modality and also the associated image analysis challenges. So in case you haven't been following the chat, I think Theodore made excellent job at answering a lot of sort of more general questions based on what the limits are on resolution, sensitivity, and how applicable it is to tissues and how to correlate maybe to transcriptomics and functional imaging. But I'm sure there's maybe also other questions. So please, if anyone wants to raise their hand or just unmute yourself and speak up. And maybe while people are still thinking about it, I had one question about sort of maybe more general, following up on what was sort of mentioned in the chat. And Theodore, for the understand is, of course, if you want to target tissues, the resolution is at the moment still like super cellular, right? So it's larger than single cells. But could you sort of envision maybe doing a bit more content aware or image content aware data acquisition, where you don't sort of densely raster scan over your sample, but you pick particular cells or tissue components from which you then acquire the data?
Yes, yeah, thank you, Robert, and thank you everyone for the questions. And if you have more and there will be not enough time, please definitely reach out to us. And answering your question specifically for tissues, indeed, look, so right now our pixel size is 5 to 10 micrometer. So as we know from microscopy, right, if we want to have a feature to be robustly resolved, we probably need to have something like 4 pixels for it, right? 1 pixel is not sufficient, in particular because our technology is relatively noisy, more noisy compared to, say, regular light microscopy. And then if we're talking about a feature of 2 by 2 pixels, which we're here talking about something which is 10 by 10 or 20 by 20 pixels, which definitely exceeds size of a cell when we section through it in tissue, right? That's why right now it's really not really the best fit of this technology and to analyze it for tissues. In particular, because spatial resolution is getting increased and increased like with every year. And we expect that next year already will be available 1 to 2 micrometer pixel size. And we even have some in-house developments supported. Mm-hmm. So that's why once we have pixel size of 1 to 2 micrometer, then it will be the time to start doing this analysis in tissues. Mm-hmm. Because everything else is possible. We can take cells, we can segment them in tissue, right? And we can start assigning pixels to individual cells. Yes, of course, we can do it. So, and we have done it, just at this point, it's near single cell resolution. Now, speaking about what you said about sort of the data-driven or microscopy-driven data acquisition. Yes, in principle, this is possible. And we constantly debate it. And pretty much every person who joins our team, he's like, oh, they are like, why not to do it this way? So exactly, because technically it's possible. However, there are still benefits of doing this in an unbiased manner. And these benefits, they spread from many, many aspects. In particular, someone, I think Julia in chat, they were asking, hey, what about background? What about those molecules which are outside of the cells, right? So actually sampling areas outside of the cells is valuable also for quality control, for detecting this background and some other technical things. And if you're interested, this problem of pixel to cell deconvolution and how to use in particular background, we have just got a preprint. It's actually in the chat box over there. But for example, Volker, he has another opinion on this matter. He always kind of tries to motivate us to do this microscopy adaptive, and maybe we'll look into this because indeed, in particular, it will help to save a lot of acquisition time, right?
Exactly, the throughput I could imagine you can add.
Yeah.
Mark, please.
Yeah, I was kind of curious, what does the raw data retention look like here? Do you need to save the ND2 and TIFF files, or do you just need to save the registered SAR files?
For the workflow, we only work with the SAR files now. I mean, people might want to keep their ND2 file. We also don't automatically delete the TIFF files in the workflow because there's not that many viewers yet for this SAR file, especially with the extensions that are not standard. We're happy to accept once there's a reference implementation to just use the standard for transformations. But yeah, if they just want to drag and drop it into Fiji or some other tool, or maybe into PowerPoint even, then it's easier for them to also have the TIFF, but we don't need it for the workflow.
Right, thank you. Christopher.
Hi, great talk. Thanks for really neat intro to these nitty-gritty registration problems. You did mention sort of these aspirations. Once physically speaking, you can reach that 1 micron target to start doing the single cell analysis in tissues. But I'm curious to hear a little bit more on how you would actually segment cells in that context. Right now, the imaging modalities that you're showing on the single cells, it does clearly resolve their boundaries, but such a problem with that bright field modality might not might not be so easy once you're talking about cells inside of tissue.
Yeah, well, for this high throughput, we've basically been just relying on cell pose in the sense that people don't need to tune a lot of parameters. We also have a tool to actually, I mean, cell pose 2.0 with the training in the loop has facilitated that a bit, but Alberto also has something for retraining for more challenging samples. I mean, there are some examples where cell pose is quite good at tissues, but it depends. I mean, if you have some really elongated cells there, you'd have to follow the literature, what comes out. I mean, there's often very specialist segmentation things appearing, but I think it's going to go all in the direction of deep learning and training suitable models and then kind of taking something from a model zoo.
And one more point here, actually. So for tissues, I posted in the chat box that we actually tried to do it. It's kind of near single cell resolution right now because of limitation discussed. But what we have done there, we took the tissue section post-MALDI and we stained it with H&E, which actually helps to see the cells and cell boundaries more pronounced. And this definitely worked much better than doing it just on bright field. So, and this was helpful. Yeah. Mm-hmm.
Sure. I guess, I guess there are some cases where H&E can clearly delineate sort of membrane interfaces, but in general, you would need like a surface marker to really resolve that at the level needed for digital segmentation. And seeing that this, the determination of what comprises a single cell in your approach relies on segmentation. I could see that actually being quite a complex problem. Now, this is not unique to sort of multimodal things where imaging mass spec is part of the loop. But everybody wants to see cell boundaries all the time. So I think there's probably a lot of other tools there, but I'm curious to see, where that takes you.
Yeah, 100%. Again, we now just stepped into this, tried a few things. At some point, we also tried phalloidine and some other stainings which are specific to cell membrane or cell boundaries. So again, so we haven't started really doing single cell in tissues. We are waiting for this new mass spec, and then we'll definitely have a look. And the field is developing so fast. We'll definitely try to take the best practices and use it for you. And maybe in the last minute. So I think Volker kind of like pitched it a little bit, but I want to emphasize it. So as you have seen, like a lot of the software, it is very field specific. And it might be, you guys might be curious how we use this and another tool, but it may be not really directly applicable. But I want to emphasize that there is a number of tools there which might be useful for the community. And this can be, in particular, with respect to the format, like this OMIZAR and ZAR in general, and ngff. So this is one. Then also, we're doing some work on developing additional helper tools for cell annotation. And right now, this is with QPath, but we're considering, for example, to re-implement it using napari. So, and there are a few other tools which we think might be useful for the community, which is much broader community than just single cell metabolomics. So if you guys found, and you've seen, Volker was actually posting, sharing a few links to GitHub there, and you will find it in the recorded video. But my point here is that if you guys will find that any of these pieces of software would be useful for you, or you think it would be, the community would benefit from this, please do reach out to us, and in particular to Volker, and we'll figure out the ways, how to publish them properly and how to really give it back to the community. Because once again, this project was really enabled and supported very strongly with the format developments, with the napari development, cell pose, and so on. And if we can also like share something back, and I think we got a few things nailed down here, in particular with the effort of Volker and Andreas, but also them being supported with others, we'll be happy to do so.
Sounds great. I think there's a question about the desktop, the Jupyter Hub setup by Conrad there. We were pretty impressed with that. How have your users experienced that? Did they like that approach? Does that work?
Well, I'm hard to speak for the users. I think generally it works quite well. Some people complain that you can't copy and paste into the VNC stuff, I have actually done a little write-up on how you build these containers, how you deploying them on a Jupyter hub. Then you need some Kubernetes people. I mean, I talked to our bio IT people. They were acknowledged in the last slide, but I'll try and find that link. Yes, Jupyter Lab desktop. So there is. Andreas already answered some. I have a specific thing of how we build with Napari in the Docker container. I have some examples for that also in GitHub, but I don't have the link here. If I don't know, is someone going to copy and paste the chat window? Then I can follow up with the names who asked. Yeah.
We could certainly make that happen. Yeah, so I mean, the alternative maybe would be, could I set up a Conda environment to run the software as well, or isn't that quite set up? Is it difficult to integrate?
So I don't think I have that slide here. I have it in an internal group meeting. So what it is, yes, you can easily run it in a Conda environment too. We package more than that in our desktop environment. For example, there is a step where we convert the raw MALDI data from the instrument to, first we split it and then it's converted to IMZML. And that is using some vendor-specific DLLs, Windows DLLs. So that is actually in a Wine Windows emulator environment within the container.
Wow.
And so we also have a Napari tool for that. And because we have everything in the container, everything is already in the right path. The users don't need to configure, you know, this environment variable. Where is this conversion tool that runs in the background? It's all there. So that is makes it very convenient. And it's all built from the CI. So, but if you just want to, I mean, you saw it on my development laptop. But what we have, the CI builds Python packages. Unfortunately, partly because we haven't stripped the parts that Theo wants to use for his commercialization. We haven't separated them clearly from the parts that we want to release open source. That's something that we hope to do. They're not on PyPI. They're in our internal container repo. So you can do pip install with our container repo as an optional argument, and then it will pull them from there in your Conda or pipenv environment, and you have all the dependencies satisfied. And again, I should point to Andreas. He's made a lot of this continuous integration magic happen.